After studying this module, you shall be able to know what is soil and how it is formed, because it is very important that if you are getting some soil on the crime scene, either it is a part of the some victim's shoe, or it has been there in the car which has been found at the crime scene, what is the origin of this soil will give you very important clue. So we will be studying about the soil and how it is formed, what are the different soil horizons, what is the evidentiary value of soil, what are the physico-chemical properties of soil which will be helping in the analysis, and then based on all this knowledge you shall be able to know what are the different methods which are used for the forensic analysis of soil. Naturally occurring soil is a complex and changing mixture of decaying organic matter, living organisms, minerals, weathering rocks and decomposing plants. So it is a complex mixture of all of these, and obviously from region to region the composition will be varying. It is a natural body divided into horizons of loose, that is unconsolidated minerals, and organic matter of variable depth on the earth's crust, which is capable of supporting plant growth. Soil can in general be considered as the natural accumulation of the humus, sand and gravel. The formation of soil is actually a dynamic process influenced by multiple factors like climate, geologic parent material, biological activity, relief and time. Soil may develop after being deposited by water, wind, human activity or animals, or even in situ. Soils are usually formed by the decomposition and disintegration of rocks and of the vegetation and animal fossils embedded in them. Rocks contain innumerable minerals which on weathering give soil particles in which various organic minerals from animal and plant sources get mixed together and form soil which is usable for different purposes. A soil is a 3D, that is three-dimensional, transferable natural material which is found on the earth's crust, and it provides natural medium to plant growth. Their properties are due to the persistent actions of climate and constituent living organisms. Soil is formed from rocks in two stages. Firstly, that is the decomposition of parent material by weathering of rocks, and secondly the development of soil profile. And we will be discussing more in detail about the soil horizon and then some cases where the soil will be found and how as a forensic scientist you can do the analysis. So let us study this in detail with the help of graphics and visuals.
Soil horizon. A soil horizon is a parallel layer to the soil surface whose physical characteristics differ from its adjacent layers. Each soil type is generally made up of 3 or 4 horizons. Horizons may be defined on the basis of obvious physical features, chiefly texture and color. These may be described by one of the two approaches, that is, in absolute terms, for example, particle size and distribution for texture, and in terms relative to the surrounding material, that is, sandier or coarser than the adjacent horizon. Layers of soil. Soil is generally made up of texturally and visually distinct layers. But from forensic point of view, generally only two top layers are important. So we are discussing here only those two layers. O, or the organic matter, that is, the superficial organic deposit with little layer of plant residues in relatively non-decomposed form. And next is the surface soil, mineral matter mixed with organics, the layer of mineral soil with soil life and the accumulation of most of the organic matter. This layer is depleted of aluminium, clay, iron, other soluble constituents, and organic compounds. Although there are a vast variety of soils in this state, each type may exist for a few yards or a few feet with a large amount of variation even in a single soil sample. Hence, to exactly pinpoint the origin of a particular soil sample is often not possible, but it is rather easy to relate it to areas of its occurrence. Now moving on to evidentiary value of soil. Soil varies laterally, that is, across the land surface from one place to another. These changes may be abrupt, occurring even within a few meters. As a function of depth, nature of soil also varies vertically. Soil changes are also sensitive towards human activity and nature. Debris originated from industrial operations and human habitation form an integral part of soil. The latter type of debris, example bricks, fragments, cinder, chemicals, paint droplets, roof shingle stones, paint chips, glass, fibers, or other items, if sufficiently unique and varied, can be most valuable factor in determining the individuality of a specimen. Soil samples with such unusual features can prove to be excellent physical evidence and may strengthen a link between specimens consequently. The composition and character of soil not only varies with depth but also laterally. In most of the cases, generally, the superficial layers of soil are examined unless there is crime like digging of a grave. However, the changes of texture and color of soils are not that much morphologically visible along the ground, but its chemical composition can show a variety of changes even in a very short distance range, and it helps in accurately determining the source of the soil sample. Therefore, in order to determine the normal distribution of soil of a particular crime scene, sufficient soil samples should be submitted. Next, moving on to impressions. Ensure that impressions in soil, such as footprints, tire marks, or any other patterns, are photographed by keeping a scale nearby to take the dimensions of that print, and a cast of plaster of Paris is made before in any manner disturbing the footprint. Photographs should be taken with the camera keeping it perpendicular to the impression. This will prevent the photograph from angle distortion. If the soil is firmly affixed to any object, it should not be removed. Such objects must be air dried and properly packed in a paper bag or other an appropriate container, along with proper sealing and labeling. Clean paper envelope should be used to pack loose sand or soil sample, which can later be sealed in a suitable container and labeled as to source. Now moving on to sample collection. Due to the nature of the soil, it is mandatory that the exact location of the collected soil sample should be properly documented by the investigators. Sites of specimen and evidence collection could be best illustrated by hand drawn sketches or detailed commercial maps. Question samples, which are generally taken from ground, like samples taken from the gate pattern of a shoe, should be compared to the control samples collected from the similar places, that is, law of comparison. Further, because the factors affecting soil formation are time governed, timely collection of evidence is also very important to ensure that examiners have adequate information of the variability of soil. A sufficient number of controlled samples should be collected from the crime scenes, along with the surrounding areas. Establishing the resemblance of a particular sample to a specific location greatly strengthens the association between them, excluding the possibility of others. Of course, the available amount of suitable soil can also limit the level of significance of comparison. In any case, the amount of question sample available for analysis cannot be controlled, but the investigators can substantially control the number as well as amount of known specimens collected. All samples should be dried before packaging, sealing, and labeling. Moist samples should be air dried at room temperature for overnight before packaging. Along with this, the collection of controlled soil samples should not be overlooked. Controlled samples should be collected from the area that could be claimed as a probable source of questioned soil. A suspect may, for example, claim that soil collected from shovel that was used to dig the victim's grave actually came from a garden. On forensic analysis and comparison of both the samples, the dissimilarities of both these samples can eliminate the yard or garden as possible sources. Next is question or suspect samples. A tablespoon of soil may be sufficient for a soil comparison. However, if the question sample is available in a larger quantity, then the controlled sample should be collected in equal quantity. The larger the quantity of soil sample, the more specific will be the comparison results. Control or comparison samples. Obtain samples consisting of at least 3 tablespoonsful of soil from each area which are suspected to be associated with the scene of crime. Controlled samples must be representative. If, for example, a pair of suspect shoes are recovered bearing a soil sample, the area of the footprint will correspond to the soil location on the shoes. While forensic soil examinations can yield important information concerning crimes and can establish the absence or presence, whatever it may be, of a link between criminal, victim, and the place of occurrence, proper handling and collection of the evidences by the investigators definitely affects the successful results. By understanding the vulnerability of earthen materials to contamination and by adopting the suitable collecting and packaging procedures, the potential forensic value of soil as an evidence can be maintained by the investigator. Next, moving on to physicochemical properties of soil. The physicochemical properties of soil are mainly attributed to its composing particles, which vary greatly in their sizes. The soil particles have been divided into various groups, which are termed as separates. Following are some important physiochemical properties of soil. First, soil texture. The mineral component constitutes the largest volume of soil mass and plays an important role in determining most of the physical characteristics of soil. The relative percentage of different soil separates sand, slate, clay is called soil texture. Next is soil structure. The soil structure is described by shape, size, and grades of soil particles. Next is soil density. Density represents weight of soil per unit volume as gram per cubic centimeter. Degree of compaction and soil makeup greatly influence the density of the soil. Soil pH, the nature of soil, that is, its alkalinity or acidity, is determined with the pH of soil. Next, moving on to soil color. Soil color is inherited from its parent materials. Often, soil forming process also influences the color of the soil and is termed as acquired or organic color. Analysis of soil. Soil is a complex mixture of both inorganic and organic materials. During a soil analysis, the class characteristics of several soil samples from a known source are compared with those of a question sample. Just as with all items of evidence that simply contain class characteristics, exclusion is possible when the class characteristics of the known samples do not match the question sample. If the crime scene location is not known, a question soil sample may hold information associated with its location. A trained forensic chemist familiar with the local geology may offer assistance in identifying regions containing soil with similar class characteristics that may potentially be the scene of crime. When soil samples and related items are forwarded to the forensic science laboratory, qualified examiners conduct a soil examination and compare two or more specimens in order to determine if the soil can be linked to a common origin. Laboratory personnel conduct the examination by comparing the texture, color, and composition of the question and control soil samples, as these characteristics are an outcome of locality dependent factors and are sensitive to a number of factors. Deviation in the characteristics tend to dissociate two samples under examination. Therefore, sufficient number of samples along with the proper documentation and collection procedure greatly increases the chances of associating soils to a common origin. This, in turn, can prove to be a valuable forensic evidence to associate or dissociate suspects with a particular scene of crime. Next, moving on to microscopic analysis. A soil analysis begins with a visual examination of several known soil samples. The natural variation of color, texture, and general appearance are recorded. The color of the sample must be analyzed when it is both wet and dry, and all visual similarities and differences must be recorded. The stereoscopic microscope magnifies the microscopic surface features of the sample, aiding in the identification of vegetative matter like leaves common to the area, paint chips indicating soil found close to a barn or house, sawdust, bone chips, or any other minute items which are not even visible to the naked eyes. This is achieved by illuminating the surface of the sample with the reflected light of the microscope. A polarized light microscope is helpful in analyzing the internal features. This process involves oscillating electric fields of plain polarized light that propagate in a single direction. The light is produced by introducing a special filter called polarizer in the light path between the light source and the sample. As the plane polarized light passes through the sample, it interacts with the sample in ways that can be measured through careful observation. For example, uniform lattice structure is a characteristic of crystalline solids because the minerals found in soil are frequently crystalline solids. This microscope can be used to determine mineral content, which in turn serves as a class characteristic of the entire soil sample. Next, moving on to size distribution. Because the individual particles in soil samples vary widely in size, it is often troublesome to determine the complete size distribution of a sample through a visual or microscopic analysis. In this case, the soil sample is made to pass through a series of sieves, that is, containers with a mesh or screen bottom, where the size of the mesh on each sieve differs. They are stacked together so that the sieve with the coarsest mesh is on the top and on the bottom is the finest mesh. The entire stack is manually or mechanically shaken for several minutes. The mass of soil retained in each sieve is then compared to the total mass of the sample that was introduced into the stack. The distribution of soil mass due to particle size is considered to be a class characteristic of the soil sample. Next is density comparison. Density is the ratio of a substance mass to volume. Both the questioned and the known samples should be collected by following the similar procedure. For example, if the questioned sample taken from the bottom of a suspect's shoe is suspected of originating from the surface of a muddy field, then the known samples should be collected from the muddy field surface and not dug from the ground. Density is typically expressed in units of grams per milliliter or kilograms per liter. It is a physical property of a substance that does not change with amount. Chemical and physical properties of this nature are intensive properties. Extensive properties such as mass or volume expressed independently change as a function of amount. For example, as the amount of a glass sample is increased, both its mass and volume increase, but its density does not. Density is commonly used in forensic science when comparing items of evidence such as glass, plastic, wood, and soil. However, because soil is a complex mixture composed of many substances, each substance will have its own unique density. Ideally, the forensic scientist will use a density gradient to evaluate the distribution of densities comprising the soil mixture. A density gradient can be prepared by carefully adding solvents of varying densities to a glass container like a narrow glass tube or a graduated cylinder. Often bromoform and methanol are used to create the gradient, with pure bromoform on the bottom, pure methanol on the top, and decreasing bromoform or methanol mixtures from bottom to top. Because substances of a lesser density will float on top of substances of a greater density, the gradient described is quite stable. It is important to recognize that substances of a greater density will sink in substances of a lesser density, and substances of an equal density remain suspended in solution. If an unknown soil sample is poured into a density gradient, each substance comprising the soil sample will sink until it reaches a region in the density gradient where its density matches that of the gradient. A visible profile of the distribution of densities of all substances found in the soil sample is obtained in this way. The profile of a question sample can be compared with a known sample to determine soil type. pH comparison. pH is defined as the negative logarithm of hydronium ion concentration in an aqueous solution. When mixed with water, many of the compounds within a soil sample will produce hydronium ions. The amount of hydronium ions produced depends on the composition of the soil. Soil pH is commonly determined by forensic scientists during a soil comparison analysis after moistening the soil with a predetermined amount of water, often 1 part soil to 1 part water. The pH of the water extract of the soil is tested using pH indicators, litmus paper, or a pH meter. Soil pH is a class characteristic used for comparison.
So dear students, let us summarize what we have learned in this module which focused on soil. Naturally occurring soil is a complex and changing mixture of decaying organic matter, living organisms, minerals, weathering rocks and decomposing plants. It is a natural body divided into horizons of loose minerals and organic matter of variable depth on the earth's crust, which is capable of supporting plant growth. We have learned that a soil horizon is a parallel layer to the soil surface whose physical characteristics differ from its adjacent layers. Each soil type is generally made up of 3 or 4 horizons that we have studied in detail. We also studied that soil samples which contain some unusual features are excellent physical evidence as they give individuality to the sample and thus strengthen the association between specimens. Due to the nature of soil, it is mandatory that the exact location of the collected soil sample should be properly documented by the investigators. The physico-chemical property of soil is mainly attributed to its composing particles, which vary greatly in their sizes. The soil particles have been divided into various groups which are termed as separates. We also studied the stereoscopic microscope magnifies the microscopic surface features of the sample, aiding in the identification of items which are not visible to the naked eyes. However, a polarized light microscope allows the analysis of internal features of soil as well. We also studied the density of soil is a very important tool to establish a link between two samples. It is measured by using density gradient tube technique. So dear students, I hope with this module you have got sufficient idea about the soil, how the soil profile is there, the soil horizons, and then how to do the soil analysis as a forensic scientist. Thank you.